Hello students, today we are going to learn about introduction and objectives of plant breeding. I am Mrs. Elrica Frida de Costa, Assistant Professor in Botany from the Government College of Arts, Science and Commerce, Kepe, Goa. The outline is Introduction to Plant Breeding and Objectives of Plant Breeding. Moving on to the learning outcomes. It helps in understanding the concepts of plant breeding and also explains the objectives of plant breeding. Introduction. Let us see what is plant breeding. Plant breeding is the science as well as art of improving the genetic makeup of plants in relation to their economic use. It consists of principles and methods that are required for favorably changing the genetic constitution of the crop plants. Let us see some brief history of plant breeding. This plant breeding was regarded to have passed through 5 distinct phases. The first one or the first phase is domestication, second is selection, the third is hybridization, fourth being novel genetic variation, and the fifth is genetic engineering. Moving on to the first phase, which is domestication. This phase is regarded as the most basic method of plant breeding because all other breeding methods become applicable to a plant species only after it has been successfully domesticated. The second phase, which is selection, was practiced mainly to exploit the genetic variation that is existing in the crop species, which resulted in a number of improved varieties, especially self-pollinated crops. The third phase, which is hybridization, the main feature of this phase is the planned and controlled hybridization between diverse selected genotypes or varieties to create new and desirable combinations among the existing genes or alleles. Now, the hybridization is further divided into 2 types, that is distant hybridization, somatic hybridization. So first, let us see what is distant hybridization. Distant hybridization is between 2 distinct species belonging to the same genus or different genera. It is mainly employed for transfer of new genes, especially for resistance to biotic and abiotic stresses from related wild species into the crop species and of alien cytoplasm with a view to generate cytoplasmic male sterility. Then somatic hybridization, it involves the fusion between protoplasts of 2 different cells. It extends the scope to distant hybridization to sexually incompatible species combinations. The fourth phase we have is novel genetics variation. Now this includes induced mutation. Now what are induced mutation? Here, new alleles of existing genes were created by inducing mutations either using radiations like X-rays or gamma rays or by using chemicals such as ethyl methane sulfonate, etc. The second type is somoclonal variation. This is the genetic variation which is present in cultured cells in plants which are mainly regenerated from them or in the progeny of such plants, and it achieves the same results as mutagenesis. The third is polyploidy, which is attempted mainly to exploit the beneficial effects of increased number of copies of a single genome or of bringing together 2 or more distinct genomes. Moving on to the 5th phase, that is genetic engineering. This phase mainly aims at isolation and cloning of specific useful genes of various organisms, their transfer and expression into plants. Now, plant breeding in India was first started in 1871 when the government of India created a department, that is the Department of Agriculture. Between 1901 to 1905, there was establishment of agricultural colleges seen in Kanpur, Pune, Sabor, Lialpur and Coimbatore. The Imperial Council of Agricultural Research was established, which was later on changed to the Indian Council of Agricultural Research in the year 1946. There was establishment of the Indian Central Cotton Committee in the year 1921. The year 1956, there was a project for intensification of regional research on cotton, oilseeds and millets. In the year 1957, All India Coordinator Maize Improvement Project was established. And in the year 1960, the first agricultural university, that is in Pantnagar, Nainital, was established. Let us see what are the main objectives of plant breeding. Now, plant breeding mainly aims at improving the various characteristics of plants so that they become more desirable agronomically and economically. Now, some of the most important objectives of plant breeding are: it helps in yielding or having higher yields, improved quality, disease and insect resistance, change in maturity duration, agronomic characteristics, photo and thermo insensitivity, synchronous maturity, non-shattering characteristics, determinate growth, dormancy, varieties for new seasons, moisture stress and salt tolerances, elimination of toxic substances. Summary: plant breeding is the art and science of improving genetic makeup of plants. It involves 5 distinct phases and it plays an important role in improving the quality of plants for agronomic as well as economic processes.